want to talk about GradioBio, which is a tool that is still in development, but wanted to give an overview and also use this as an opportunity to get some feedback, maybe so what kind of features could be useful. Okay, and just to mention, I'll try to mostly not talk about pipelines today because we've seen kind of some pipelines in the earlier iterations of this series. So I want to speak more about the kind of standardization of models and how can we make different models talk to each other without going all the way to the pipeline. Why I'm doing this, so maybe some of you are familiar with these apps that I've been releasing. So this is, I think, arguably the most popular one for ProteinMPNN. It's running on Hugging Face, and it's a simple web app, so you can kind of enter like some 1 PDB code. So let's take, for example, 6mmR, and then you can just click a button, and in your browser you can run ProteinMPNN quite easily. And then there's also structure validation using AlphaFold and so on. So this app was quite popular, and it's also super easy to use for kind of non-computational people. So if you just structure the protein, you work in a lab, and you maybe want to try get some mutants, and this kind of really reduced the barrier by quite a bit. And I also got some feedback from people using this for teaching and so on. And the idea was, now that the field has matured a bit, we have many more models. The idea was, how can we standardize this a bit more so that kind of more tools are available in this type of fashion? So what I want to speak about is basically briefly talk about how people interact with AI models in general, because I think it's relevant to what I will show afterwards. Then briefly talk about the landscape as I see it, and then also kind of the challenges that I see. And then afterwards, introduce you to this GradioBio library to show some of the things that I've been building so that these things get easier, hopefully, in time. So, and kind of if we talk about AI models in general, right, then most of you probably see one of these screens in their day-to-day workflow, right? So we have these prompt interfaces, and we have these kind of agentic interfaces that live in your terminal. But the main thing is that if you interact with it, the model is not running locally. It's mostly running in a cloud somewhere. And if you want to build an application, the first thing you do is get an API key to actually access these applications. But then this is in general. So if you look at our field, at biology, protein design, protein structure, prediction, and so on, and we've seen that this has also happened a little bit. So like on the left, I'm listing quite a few of these companies that kind of offer these kind of services that are similar to this ProteinMPNN app that I just shown you. So it's kind of a website, you register, you might have to pay them some money for the compute that you're using, but then afterwards you have a web form, you can enter the backbone you want to analyze or the sequence you want to predict, and you click a button and then you get the output. But then if we think about it in the perspective of an academic, then most of these platforms we can't really use, because actually for most of us, we have access to kind of this subsidized academic compute, right? We have our workstations, we have some cluster that the university maintains or that we can apply for and then can use it for free. These kind of cloud providers are a bit off limits to us, but it would be nice if you would have something like this. So I'm sure if people are familiar with this. This is Brian Norton who maintains this biomodels repository, which has quite a few of the popular models, so from ProteinMPNN, LigandMPNN, different structure prediction methods, and so on. And you can all use them on Modal, which is also a cloud provider for compute, but in a serverless fashion. So basically, you run this single script, and you can directly run the model, and then you get the output locally, but it's running in the cloud. But obviously, it requires paying this compute provider for the compute. So ideally, we want something that is as convenient as this, but that kind of allows us to use our academic compute platforms, and also that allows us, that is very flexible and allowing us to chain together different models. And that could also then facilitate something like this. So I'm not sure if people have seen this, or there's this protein-based website from AdaptiveBio where they host the data from the competitions that they've been running, but they have this kind of skills page that they set up. So for example, you could, if you install these skills and Claude or any other agent that has skills, they can run these models by running these. Click on ProteinMPNN, for example, then you see somewhere here it's using this biomodel interface to figure out how to run the model. So what the challenges that I see is, so what do we use to make these models talk to each other? How do we ensure that the models are portable and resistant to time, right? So I'm not sure if people have tried to run, for example, ESMFold on modern, like the recent CUDA hardware, but at least I get some errors with this. So we also have this kind of problem that scientists really want our things to be reproducible. Maybe if it's 2030, I still want to be able to run ESMFold, and then ideally I should not have to figure out how to make it install, but ideally I can reuse some portable install instructions that kind of work still after a few years. Then more recent challenge is also how do we use these models together with agents, because they are really quite helpful in doing our research. And then I think the biggest challenge is actually how to get people to adopt this, because everything that you put on top of actually training and developing and releasing the model is extra work that not everyone might want to do. And that brings me to this XKCD, right, where we are, I'm not sure if someone has already shown it, but this is the risk that if we're talking about a standard, it will just lead to there being more standards. This is something that we should be conscious about to not reinvent the wheel just to reinvent the wheel, but try to go in a direction where people can get behind in actually adopting it as well. Therefore, finding the minimum standards that everyone agrees of is sensible to actually do. And ideally, we would want to go somewhere like this. So another XKCD cartoon. So obviously, this is a bit hyperbolic, right? This universal converter box doesn't really exist, but ideally, we want something like a program that can take any of the outputs of the models that we have, and then you can use that output and put it into a different model to process. So this is the aspirational goal of where we would want to go. And if we look at some of the tools that have been presented so far, right, they've mainly been in this pipeline format, where we have some orchestrator program that kind of calls one program after another, and then makes them the input formats work. And that kind of then is quite a bit of development work to basically make sure you're tracking all of the different settings and parameters. And if you want to add a new model, it is like people have to get familiar with the syntax that you're using. And the best developed standard is Nextflow. It comes more from the bioinformatics pipeline, but there is also some protein design or structure prediction methods that are available in Nextflow, which have really, they've done most of the standardization work already, right? Where you can write these Nextflow plugins in different programming languages, and the data flow is defined, they containerize the applications, and then you also have version control, and afterwards it's this runtime environment, so you can run things on different platforms, and that can be platforms that people use in industry, like AWS or GCP, but it can also be platforms that kind of academics use, like Slum Schedule or on some kind of supercomputer. But as we can see, right, the adoption of this for our community is pretty minimal, because it's a time effort to actually make your models available in this, and then afterwards it's a bit rigid in how you can make things talk to each other and combine them in a pipeline. And kind of the other extreme is where you have some reusability by people by using these Google Colab environments. The only thing that you have to do is, apart from writing your Python program, is you have to add these little kind of comments to define what kind of input parameters your model takes. And then this has been quite successful at this Colab Fold platform, for example. But also over time, I think people have realized that it's not super maintainable, because Google uses the same container image for all the Colab instances. If you try to run something that someone put out there maybe 2-3 years ago, the chances are that you will have to tinker a bit with the packages. If the people have not been maintaining it actively, it's quite hard over time, just the computational environment changes. So it's nice, but it's also not ideal. So to come back to these challenges, what I see from my perspective, or what I want to do, is basically, so in order to make models talk to each other, I don't think we really need like a big orchestrator pipeline engine or something, but we really should just have small API interfaces that the models expose, and then have some standardization for the in and output formats that these models accept. So for example, if you want to run a sequence design job and you use ProteinMPNN or another sequence design model, ideally you just have to change the key for which model to use, and then afterwards you get the prediction of the different model, but you don't have to actually change the format of the input, or the one takes a YAML file as input and the other maybe uses some bash arguments, stuff like that. So here we should have some standardization. For this kind of portability and resistance to time, I think the lowest common denominator that most people, I think, already agree with is using it in containers. And if we have an API, that we can also use this model context protocol, so that an agent can understand what is happening. And then basically the idea of how people can actually adopt this is that we start by introducing as little overhead as possible. And this is basically where I think the sweet spot is, which I want to talk to you about, is this kind of Gradio tool, and then this GradioBio library that I'm developing that kind of hopefully makes things a bit easier for us bio people using it, because Gradio is a kind of a general purpose framework developed for machining. So my vision is that basically all these tools, they live in different isolated containers, and then basically if users or agents interact with them, they do this via kind of a unified API or using UI. So basically you can both use it in a kind of testing fashion. Like you can use this kind of ProteinMPNN web app where you just go to the website, you enter your commands, and then you can try the output. But also, if you really want to use the model, you take this container, you run it in some place of your choice, for example, your supercomputer or your workstation, and then you can interact using the API with it. And in that way, it's very similar how people interact with these kind of commercial AI applications from Google or OpenAI, because you do all the communication via HTTP. And so it's very portable because you can run multiple containers, not necessarily the same machine, but it can also be on different machines. So this is what I want to do with this GradioBio framework. It's still work in progress, so it's not completely finished, but I can give you an overview of what I've been implementing so far in the next 15 minutes that we still have. So just as a quick basis, what is Gradio for those that don't know? So Gradio is basically a framework of which you can build interfaces, and it's pretty simple. So if we look at this code, this is all what is needed to build this interface here on the right. So you define certain blocks. So for example, here a text box block, or some special blocks like this a molecule output. And then there's a run button that you can define, and then you can attach an event listener to this run button that defines which Python function should be run and what the input and what the output is. And then in this Python function, you can really do anything you want. It's just a standard Python, and in the end, you just have to output what the output component expects. So for example, this molecule 3D component takes a path to a PDB file or a zip file to kind of display a structure. And this Gradio framework is nice, and I like it much more than some of the other alternatives because it has very minimal setup and it's quite intuitive to use. For example, if you compare with Streamlit, which is quite similar in functionality, but for example, they are only now introducing a REST API, which is the key feature that is needed to be able to talk to these models programmatically instead of no one wants to click for actual design workflows in the browser on different buttons. And then there's some kind of older alternatives like Shiny from the R ecosystem or Flask, which kind of gives a more advanced web server. So Gradio is just it's a good enough abstraction to make it easy to build these applications, but it's not blocking you in any way, but then it's also quite easy to use. And it's really just a few lines of Python to turn the script that you use to run inference, which has maybe some argparse arguments. And if you have this, it's very easy to turn it into an application like this that then has quite a few nice features. The first one is that basically you automatically get this API interface. For example, for this ProteinMPNN app, if you want to use it using an API, you install this client library, but you can also do it using just normal CURL or like HTTP requests. And then you initialize your client, and then you can run predictions by providing the same input argument. So here, for example, I have this input sequence. If I go back here, just this input text box, and in the API, it basically looks the same. And then if I get the result, it will print me the PDB file. And there's a lot of nice features. So basically, by default, these Gradio apps, they support streaming output. There's a queuing system. So for example, if you have one node that is doing all your ProteinMPNN jobs and you submit hundreds, then automatically there will be a queue and they will be processed one after the other, or you can also set some concurrency and so on. This is all integrated. And most importantly as well, the whole framework is open source. So it's not dependent on someone abandoning the project, and then all the effort that we put in it is basically lost. This can happen with some Google products, I've been told. And afterwards, the idea is if whatever other application you have. So, for example, we heard about this Rosetta Link approach, where I think it was Ayaza who presented this, right? Where you want to have Rosetta movers that interact with ProteinMPNN, for example. Inside these scripts, you can just then call the API endpoint, and you can basically do all the communication between your model and whatever other program you have using the web, and that's super general. So you can do it remotely, right? So you can run your application somewhere on the supercomputer or on your workstation. You can still work on your laptop and get the results there because you can communicate over the internet. So really quite nice feature. And then the cool thing is also that they have this API recorder. So here in this animation, you basically see that there's an app that has multiple steps. And on the bottom left, there's basically, you can click on record. You can, in the user interface, run all the things that you want to do. And then afterwards, you get the code, how to do that programmatically. So it's the barrier to entry for people that don't program a lot is quite low. So they can construct the workflows that they want by simply chaining calls to different applications that they run. And then another important feature is discoverability, right? So you don't want to create something where afterwards people can can't find it. And these Gradio applications, they really work great in the current ecosystem that we already have. So you can launch these apps inside Google Colab. They can be hosted on this Hugging Face platform for free, like this ProteinMPNN app. So on Hugging Face, you can host models. This is not where this would go. You can also host datasets. But the third kind of platform is this Spaces, where people can host interactive, they call it demos, but it's a Docker image in which you have the code, how to run your model, and some resources in which this model is running. And the great thing is that with one command, someone can basically take your application and run it locally because they have a Docker registry already set up. So basically, for example, if you want to run this ProteinMPNN space that I've been showing you in the beginning locally, then you use this command that you can copy from the page, and then afterwards you have it running locally on your own infrastructure. So using this Gradio ecosystem allows us to basically profit from some of the effort that Hugging Face has invested in making these models accessible. And another cool feature is this kind of link sharing. So basically, they also have a tunneling feature where you can, if you're working in a university, most often they have a firewall which prevents you from directly connecting to the servers in the university. But basically, if you run one of these Gradio applications and you add this share equal true flag, then you will be able to bypass the firewall of university because your kind of connection goes through a tunnel, which means you can basically, so you can run something on your workstation, you can give the link to your collaborator in a completely different university, and they can directly run like the model on your resources for testing. So it's really great also to quickly test things. This kind of for the background for Gradio. And now, obviously, we need some special things because by default, Gradio is mainly intended for like standard machine learning, so LLMs or image models. So they have some components which you can use to input structures or other things, but we need something special for biology. And I built this library of components. So there's something for molecules, for inputting co-folding models, for inputting these kind of contig segments, and as well for sequences. I'm going to go a bit quicker because of time, but this is kind of the component that you can use for structure. So it can be used to visualize a structure, but then also there's quite a few options in which you can color and do this programmatically. And also people can select residues in the structure, and then you can use these selections as the input for models, for example, for selecting hotspots and so on. And the idea is always that you have this kind of, you can use it as a human using the interface, but then you can also do the same thing via the API so that it's easy to use. This component already exists, but this addition with Moldstar is, I haven't released it yet, so this will come soon. And the idea is basically also that it can take normal PDB and CIF input, but then, for example, that this input framework, which I'll present in a second, can also take as input, for example, structures from AtomWorks, which I believe is presented after this, or from some libraries like Biotite. There's also this co-folding input, which also already exists, but I have made it better. So this is the improved version where basically you can set up a co-folding job. And the idea is that basically this GradioBio library will set up this input, and then afterwards you can run using the same input you set up in this way. You can run a Boltz-2 prediction or an AlphaFold 3 prediction or a Chai-2 prediction. And so some of the features, so here, for example, I'm selecting an attachment atom. So this is a feature that Boltz supports that maybe some other model might not support. But the idea is basically to translate what is possible between the different models so it is as portable as possible. Then this is pretty standard contig input, which is a kind of a reusable component that people can use to set up their jobs. And then the same thing for sequences where you can label different residues and you can use this kind of in output of sequences with annotations. And kind of the last thing that I want to quickly speak about is basically the tracking of parameters and of the metrics that are produced by these models, right? So if we talk about Rosetta, then there's kind of this concept of a pose, which is a structure, and then there's kind of certain methods how to deal with this structure. But ideally, for these kind of AI models, we would have something similar that is not dependent on a library like Rosetta, which is great, but it's quite heavy, right? So ideally, we would want something that is in a text file. And actually, we already have this file format. It's the CIF file format, which I think many people don't like, but it has quite a lot of features because it's dictionary based. You can pretty much store any kind of information in it. And in this framework, what I'm doing is basically that I'm storing, or there's a little wrapper you can use to easily store different information in these CIF files. So like sequence coordinates, but also the design parameters or the pipeline that you use to generate a specific structure. And then also all the design metrics like the PLDT, PAE, and so on, which are outputted by some models inside a CIF file, but for some others, you kind of just get a JSON that is side by side with your structure. And ideally, you should have this one text file that has all the information about how the structure was generated and what are the different metrics that kind of come with this kind of GradioBio library will attempt to basically do this. And it builds on this model CIF standard, which is something that some people from the Swiss Institute of Bioinformatics and people from the different protein data banks and so on, they have coordinated this already. So it's already something that's in place where basically they are also storing the annotations, like what kind of model was a structure that is deposited, for example, in AlphaFoldDB, what kind of model was it generated with, and they also store, for example, the PLDT or the PAE. We just basically need an easy interface how we can store or do this more frequently for outputs of different models so that in the end, these kind of CIF files are kind of like a dictionary that kind of contains all the information about your job. And the interface that I kind of came up with looks a bit like this. So you can basically, you have this kind of flow-wise interface where you set up certain outputs. So for example, this could be a sequence generated by ProteinMPNN. And then with this easy API, you can basically add different parameters to it. So then afterwards you store it in the CIF file and you have basically all the parameters together instead of having this FASTA-like format with all the things written to the header. So this way it's more standardized and more malleable to whatever model that you're using. For structure output, it kind of looks similar, right? So you initialize the existing structure, and then you can add different parameters to it. So for example, PLDT, IPTM, or these other things. So this way you have everything in a single file that afterwards you can transfer, and then you can read it again using this library. And then this would allow applications to this one. So this already works. If I take a CIF file, it has this kind of PAE information in it, and then I load it on Mostar, for example, and then I directly have this PAE matrix that is visible. So if we stick to this already existing standard, then these metrics will always come together with the structures. And if more viewers also support this, then it will be quite useful for us when we can analyze our results, for example, or we exchange them, or we put them in some repository when we publish a paper. So to summarize, what this GradioBio library will attempts to do is basically to standardize a bit the I/O from different models by providing these input components, like this co-folding input that then afterwards has light wrappers to translate the general input into specific inputs for some initial models. And then obviously there's some programming work involved to do this for more models, but the idea is that this is just a very light abstraction. Then also to have these kind of isolated containers, so not a whole pipeline of things that you have to install the whole suite, but basically you have a single Docker container, a single command, and if you run it, then you have this API interface so you can interact with this type of model, and then afterwards what you want to do with it, or if you want to chain it together to create your design pipeline, that's totally up to the user. It's not something the library wants to do, and then basically also allow this kind of provenance tracking and kind of having this unified CIF format as the kind of dictionary that contains not only the output coordinates of your job, but then also some of the other parameters that kind of might matter. And as I said, so this is still work in progress. So the components are almost ready, and I'll attempt to basically have a release with the first methods in maybe a month or so. That will be useful. And if you have feedback or if you this, then please don't hesitate to reach out, and then I'll be happy to hear your suggestions for basically what the input components, for example, should look like. With that, thank you, and happy to hear any questions.
Great, thank you very much. You can paste the questions in the chat, you can raise your hand. So far there's no questions, but of course I definitely have many. Maybe I can start with a comment. I think having a universal pose is probably a good idea. Like Rosetta, from one step to the other does that, and then it enables you to build the building blocks much easier if you have a standard pose. So this model CIF is a great idea. I didn't know that before. And the other comment is you still had to write, or are you in the process of writing Python wrappers around RF diffusion probably, and bolts and chai and AlphaFold 3, because you're using them in GradioBio. So I think this is where we could really step together, because ProtFlow is also doing the same, Rosetta Link is also doing the same. So we could just write the Python wrappers once, and then everybody would use them for their own thing, because I don't think it makes sense to. This is an easy, most people are using Python, so most people would benefit from that. We have a question. I don't know if it's completely on topic. So how do we best implement iterative design pipelines using this kind of workflow managers?
With iterative, you mean that kind of
It's probably step by step, right? Yeah. Can you build different steps in a pipeline? Can you make a pipeline out of that.
Yeah, so in the end, all these things are just basically a tool that you can call, and the iteration you would have to do outside of it, right? So you have like your Python notebook where you have a for loop, you have that many cycles, for example, and then for each cycle you do one HTTP request to one of these models. So the aim of the library is not to write the pipeline for you, because I think that's anyway, at least from my point of view, it's a bit hard to standardize, because people, they try different optimization algorithms or these kind of things. So this is where I think you would want to play. But then the call itself to ProteinMPNN and the use of model MPNN is the same. So here, this is where I think the standardization would be helpful, so that you don't have to figure out again, okay, what are the dependencies of this package? How do I install it in my environment and so on? So you can run this one command, docker run, and then you have the thing up and you can directly interact with it. And then also in this Gradio framework, you have the added benefit that you don't only have a command line interface, but you have also a user interface. So you can directly explore without kind of having to figure out, okay, how do I run the thing? Yeah.
Great, thanks. Hey Andrew, good to see you. You can unmute and ask in person. Let's be interactive.
All right, thanks, Yasa. Really nice talk, Simon. I'm curious about that bit you mentioned about getting Docker on HPC and like getting past firewall stuff. Can you elaborate on that a bit more? I'm not sure I understand. So where can you use these GradioBio models? Can you use them on like a standard HPC system?
It depends on if your standard HPC system has Docker installed, but I mean, if it's running on Docker, you can also run it on some of these other kind of HPC container management systems. I think that's what was it called?
Singularity. Yeah, Singularity or Optator, they're more or less the same, and they're compatible with Dockers.
Yeah. If you have a Docker image, then I think that's easy to do. And then basically what this link tunneling feature is doing is that, so Hugging Face is maintaining a server that allows you to kind of tunnel through your app, bypassing any kind of firewall in between, but also this kind of server implementation is open source. So for example, if your university would want to host it themselves, they could as well. So the idea is simply that this is running somewhere, and then you get a link that you can give to anyone in the world, and they can, if they go to this link, all the commands they enter in the interface, they will end up on the host that is running this. And then obviously, there are some security concerns with this, right? Because there's no password or things like this. The Gradio people, they've done kind of like a security review. So basically, only the files that you explicitly allow your app to see, those can be actually returned. And these apps or these links, they don't live forever, but they live only as long as the program is running. And then even if the program is running, I think after 72 hours or if, some delay, that the link connection will be severed.
Gotcha. Just also, which is that. So I mean, is the models being hosted on Hugging Face? Where does the compute happen? Does the compute happen on your HPC?
The model is not hosted on Hugging Face. You take this container, you run it on your hardware, and this hardware is running the thing, but you can access it from any link.
I see. Gotcha.
And you have to check your network policy because some universities probably prohibit such opening of firewalls.
For them, it's not. Yeah. They have no way of knowing, because they track all HTTP traffic and then analyze it. So it's
a fair point. We have a technical question, which I'm also curious about. How does Gradio compare to Streamlit? Is it also rerun on each event, basically, or not? Or in general, what are the capabilities of each?
Yeah, so they do pretty much the same thing, but the benefit of Gradio is that it has this kind of quite mature API interface. And then this goes also to the second question, where Ravi asks, tools getting mainstream, does the web interface still matter as much? And yes, it does, because basically the web interface is then all the form fields or all the inputs that you have in the web interface, those are what define your API as well. So the web interface just exists because you have the API. So you have to create your web interface once, and then you have the API that directly comes with it. And that's the main difference to Streamlit, so I think until now they didn't have an API, and now since I think this January they have an alpha version where you can define certain API endpoint routes, but it's not automatic, but you have to explicitly define all the API endpoints. And with Gradio, any Gradio app that you on Hugging Face, you go on it, you scroll down, there's a button, use via API, you click on it, and then you can directly send HTTP requests and use the app in exactly the same way.